It's my great pleasure to start this next symposium session with Dr. Allan Reiss, who's a professor of psychiatry, radiology, and pediatrics at Stanford University, and a board-certified child and adolescent and adult neuropsychiatrist. Dr. Reiss specializes in the assessment and treatment of individuals with identifiable genetic, neurodevelopmental, and/or medical risk factors for learning and behavioral challenges. So we'll learn about the psychiatric view. In his research, he uses multimodal research methods to determine how biological and environmental factors affect brain structure and connectivity and function, and how this ultimately impacts outcome in individuals with typical and atypical development. Lastly, a particular focus of his work is to derive explanatory models that inform the development of more specific and effective interventions for those with brain disorders. Really happy to hear now the talk of Dr. Allan Reiss, which is titled Neuropsychiatric and Cognitive Neuroscience Applications. Thank you. The stage is yours.
Thank you, Alex. So I have an admission to make. This is my first professional travel since 2019, professional travel. So here I am in Boston. Thank you for inviting me, David, and the organizing group and the Chen Institute. I have to tell you, when anyone gives me a microphone and a power pack, I kind of go into automatic mode. One of the things I have been doing all during this time of the pandemic is teaching group exercise classes. So I told Alex, maybe I'd get you all up after the meal and do some exercise, but I won't. But I do 3 or 4 times a week. And so I was doing my own little routine, uncurl the microphone cable, get it all ready. Anyhow, it's great to be here. I promise I won't make you exercise. You know, I had a completely different talk planned for today prior to, I'd say, 10 days ago. And then David and associates sent out an email saying, well, look, why don't you really concentrate on challenges as opposed to a more traditional talk, speaking about your work and how excited you are, and which I am. I'm very excited about our work, but I want to talk more about challenges in my field and more broadly in cognitive and clinical neuroscience and how it relates to our topic today, which is imaging in the everyday world. This is our agenda for today. So we're going to talk about, with one slide only, our FNIRS research at Stanford from my lab, which we call the Center for Interdisciplinary Brain Sciences Research. Then I'm going to give you a relatively brief tour, since this is only a 25-30-minute talk, on the present state of neuropsychiatry, which is actually clinical neuroscience. You know, there are no signposts in the brain saying this belongs to psychiatry, this belongs to neurology, and so forth. We're going to talk a little bit about the human species. Challenges will be embedded throughout today's talk, and at least my take on how eventually we can address challenges. I'm not nearly as optimistic as Joy that it's a 5-year trip. I think this is a long trip, and I hope to tell you why. Okay, what do we do at Stanford in terms of FNIRS research? This is what we do. We have clinical applications and cognitive neuroscience applications. We have a number of different disorders that we study. Some of these you may not even have heard of. Fragile X syndrome. Raise your hand if you've heard of Fragile X syndrome. Yeah, that's a lot more than it would have been 10 years ago. Klinefelter syndrome. Raise your hand if you've heard of it. Autism spectrum disorder. Certainly all of you or most of you have heard. Attention deficit, post-traumatic stress. We've looked at using FNIRS to assess cognitive effects of type 1 diabetes in children and adolescents. And more recently, in a collaboration with UC Berkeley on the effects of insecticide exposure in the Central Valley of California in a very, very large epidemiologic type study on outcome. We also have a lot of cognitive neuroscience applications, which I've listed there: exercise, creativity, humor, driving, cooperation, collaboration, social, emotional interaction, effects of the virtual environment. I mentioned a couple of those studies at the end of Joy's talk. Cognitive load. Just want to highlight a couple of things. Yuan Gao, who is in the audience now, has been working with me on our epidemiologic data from insecticide exposure in Berkeley. She's also about to embark on a new ASD study looking at movement and motion in that population as a potential biomarker or as a way to differentiate subtypes within that very broad category. We have Cassie Yang, who's not here today. She wanted to be, but she had a different obligation, working on virtual reality plus functional neurs in individuals who have relatively less or more attentional problems, so kind of in the broad area of ADHD. Stephanie Balters, who I've mentioned to a few of you today, working in the area of PTSD, and is also working hard to see if we can infer deep brain activity from surface FNIRS that we validate with concurrent fMRI. Those are just some of the areas that we work on, and that's it. That's all I'm going to tell you about today in terms of our work. One slide. All right, let's talk about psychiatry or clinical neuroscience and what do the present data tell us. Well, I have to tell you, I almost went into neurology. I eventually went into psychiatry for a very specific reason. It was in the late 1970s. How many of you have heard of the DSM, Diagnostic and Statistical Manual? Okay, a lot of. So, do you know what the statistical part of the Diagnostic and Statistical Manual is? Does anyone know? What is it? It's all replication. It's a, is it reliable? Can one clinician diagnose it in the same manner as another clinician? There's no other statistics associated with it. It's really a group of so-called experts who get together and they decide on the clustering of symptoms and signs, and for childhood diagnoses, developmental sequences that tend to co-occur and that therefore define a condition. Now, in the late 1970s, when I was finishing up medical school, it was a very heady time because the DSM-3 was coming out, and it was the most phenomenologically oriented, not pseudo-etiologic oriented manual. So I thought, this is it, man. Let's get into this field because we're going to solve some of life's greatest mysteries, why people get very, very sick and why it's so common. Well, it turned out that that was not the case. In fact, I'm going to ask you this. And I've only asked this question at a couple of talks where people have correctly given the answer. So we're up to the DSM-5 now and a revised version of DSM-5. So remember I was talking about the DSM-3 back in the late 1970s. There is only one diagnosis that has ever been constructed as part of the DSM sequence for which there is a pretty specific etiology and pathogenesis. For the prize today, a free exercise class, what is that diagnosis? Does anyone know? In the back. Syphilis. Wow, that's a good guess, but no, syphilis never made it into the DSM. But it's a great guess. Really good guess. People oftentimes say Alzheimer's, which is, of course, not true. Alzheimer's is a very heterogeneous category. Anyone?
Course costs?
Addiction? Course cost? No, no, no. Anyone? Depression? No. No. Schizophrenia, good guess. These are all good guesses. It's Rett syndrome. How many of you heard of Rett syndrome? It's really, I won't talk much about it. It's a very devastating, very early neurogenetic, neurodevelopmental condition that's much, much more prevalent in girls than boys. You have normal development for a few months, maybe up to a year, and then you have developmental plateauing regression. It's really quite awful, and you develop autism-like symptoms. I took care of a lot of these girls when I worked at Johns Hopkins prior to coming to Stanford. It's really tough. It's the only one, and it was in DSM-4. And what happened to it in DSM-5? It's gone. So the DSM is really a diagnostic manual of mysterious maladies, really. It's not anything to do with specific conditions with biologic or genetic constructability. And this is what I'm showing you, is that for any DSM diagnosis you can find now, there are multiple pathophysiological mechanisms and neural pathways. And even more so, there are so many different risk factors that some of which are moderate or greater influence, and some need to be combined to eventually lead to the pathogenesis and the diagnosis. So when you think about a diagnosis like autism spectrum disorder, like depression, like schizophrenia, please think about this, because it can really mess up your experiment if you expect your results to be consistent and homogeneous, because they won't be, because the condition itself, the diagnosis itself is not. And it's even more complicated than that. Even when you have specific risk factors, and I'm listing some of these genetic ones here. FMR1 is the gene that's involved in Fragile X, MECP2, Rett syndrome. This is a neuroligin, neurexin, associated with autism spectrum disorder, and so on and so on. I was talking to someone about 22q11 deletion syndrome. That's actually associated with a 20-fold increased risk in schizophrenia, but yet we still don't know the pathogenesis there. So each of these by themselves can give rise to a whole host of different outcomes, including, in many cases, unaffected. So the reality of pathogenesis of these very, very common conditions is that they're significantly complicated, significantly heterogeneous, and we need a lot of work to try and figure that out. So this is one reason I won't focus very much on this slide, why I have pivoted, or I pivoted very early in my career to this approach called behavioral neurogenetics, where we focus on presumably more tractable diseases that have a single pathogenesis or a shared pathogenesis, and supposedly are more tractable, conduct investigation at multiple levels. But you know what, as it turns out, even these single gene disorders, these deletion syndromes, are really complicated too. It's not just a simple A goes to B goes to C. So Fragile X, which many of you have heard of, is the most common heritable cause of intellectual disability and autism spectrum disorder. And we know when the gene was first described, by the way, it was also very heady times. That was in 1981. I had been doing work in this area for a few years already. And we thought, okay, we're going to cure this. We're going to cure kids who have autism spectrum disorder from this single gene disorder, because we have a clue as to what the protein does that's in relatively short supply in the body and brain. Sorry, much more complicated as it turns out. It has a bunch of different functions, this protein, and this is just a slide showing many of those functions. So if life were easy, we'd be doing a lot better, but it's not easy or simple. So in the field, we have this paradigm, potential inflection point, paradigm shift. And the dissonance gap here is between the strategic inflection point and the recognition of the shift. And during this period, diverging ideas, practices, and approaches cause conflicting opinions. So when I would give a talk like this 25 years ago, I'd be carried out on something, out of the room. But now most people actually do believe that, in fact, we are at the strategic inflection point. We can't keep going in the same way, saying, let's just study schizophrenia, let's just study ADHD, because it's not going to lead us anywhere new. We have to have a different approach, which can, in fact, generate challenges, but it's more exciting. Because if we don't, again, we're going to have problems, and our patients and our friends and family are going to continue to have chronic neuropsychiatric, clinical neuroscience issues. That's from the New Yorker, by the way. Great cartoon. Okay, so how can we better understand pathophysiology and improve outcomes of persons with brain and behavioral dysfunction? Well, you have to understand state and trait. A lot of people were getting at this today, and some of the posters are getting at this. So state means your current condition, how you're feeling, how you're motivated, how much sleep you got. Whereas trait means, your genetic predisposition, your genetic risk factors, your stable home factors. We have to differentiate those two, because at any single moment, you can have functional brain activity that is highly state dependent and can shift to a different topography based on changing state. Remember, DSM diagnoses are not real diseases, and they're unlikely to help specify pathophysiology. We need to conduct developmental longitudinal research. The only way we're ever going to figure out cause and effect is to follow people over time. From a cross-sectional study, you can get clues. but you have to follow people over time. And of course, getting to the theme of this meeting, we need to do naturalistic studies. In-lab studies are fine. It's a great proving ground, but then we have to move to more naturalistic studies. So there have been numerous studies where you ask parents of children who have serious problems to fill out checklists, and then you have raters rate them, and there can be significant discrepancies between the two. And you need to collect multidimensional data, including brain. Alex talked about this, other people have talked about this. We need to have really robust tools for doing multidimensional analyses of these data. Uh-oh, I fixed these and they still came out bad. Okay, so we need to talk about it a lot. So congratulations to David and this group, Chen Institute, for arranging this meeting. We need to do some heavy lifting, and I'll talk about the heavy lifting a little later. We need to talk to one another. We need to collaborate. And you can't see that, but there are dollar signs there. We do need money. And I have a completely different perspective than Joy that you do about where we should get that money. As having chaired 2 NIH study sections, I don't think NIH is the way to go, NSF. It's so political, it's so complicated, it's so regulatory, it's so overburdensome at this point that I'm not sure that even a big infusion is going to make the difference. Okay. So let's pivot a little bit. And I think our first speaker talked about consciousness, right? Isn't that right today? I'm going to tell you the secret of consciousness today. But I'm not going to tell you what consciousness is. I'm going to tell you what consciousness is not. Okay, so I hope you bear with me. So let's talk about our species a little bit. So homo means human being. Sapiens means wise, discerning, and sensible. Whoops, how did that get in there? Okay. You have to remember that our species, sorry, I just couldn't resist a little political. I hope I didn't offend anybody. They're just mugshots. That's all they are. I'm not making any other comment. Our species is a very recent addition to our planet. The Earth is 4.5 billion years old. First unicellular life 3.5 to 4 billion years ago. Multicellular life 600 million years ago. Homo sapiens 200 to 300,000 years ago. Migration out of Africa 40 to 60,000 years ago. Written language 5,000 years ago. Industrialization, 200 to 250 years ago. Did you know that 300 years ago, most people could not read? 200 years ago, most people could not read. Dyslexia is a very recent addition to our diagnostic armamentarium, and it's only because we've been teaching people to do things that their brains weren't necessarily meant to do on an evolutionary basis. So my tentative conclusion is that we are truly a primitive species, given the recentness of our biological existence and developed culture. Okay, so why is that important? Well, think about the evolution of our species. This is just for fun. Okay, so there you go. That's one way to conceptualize the evolution of human beings. Here's another way. Take that in for a sec. Okay. How do we learn more about human beings? Well, right now, even with the great developments in FNIR's technology and other sensors, we still have insufficient robustness and granularity of real-time, high-resolution sensors. As I mentioned, processing and analysis of high-dimensional data still has a long way to go, particularly real-time. We need to have better definitions and at least some standardization of what the environment means. Remember state and trait. One way we understand state is to understand how the environment is affecting that state. Limited sample of cohorts and the problem of consciousness. So here are the 3 things I think are particularly relevant to FNIRS and clinical neuroscience in the natural world. I'm just throwing these in because they're kind of fun to think about. So what could our sensors look like in the future? How many of you have heard of neural dust? It's actually, yeah, David. So this was my what I thought was going to be the miracle sensor. It's developed at UC Berkeley, and it was supposed to be an injected polymer that could both measure and stimulate nerve activity. And so I wasn't necessarily going to be the first person to volunteer for this, but it's injectable. They're very, very tiny. You can have hundreds of thousands, if not more, circulating in your body. Well, it turns out these things are a little bit too big to do that. So you can inject them locally and certainly peripherally, and they can both measure and stimulate nerve activity. But something like that, measuring from cerebral organoids, so those are, of course, brain in a dish. Those are becoming more and more advanced. There are ethical issues here that are arising, which I won't get into. Okay. How do we improve our sensors and measurement capabilities? Well, we already know we can do this. fMRI, EEG, FNIRS, both single and hyperscanning. Biology, I saw some posters and I've heard other people mention it in their talks. We can measure physiological parameters. We can measure cortisol, stress hormone, sex hormones, behavior. Interpretability of FNIRS, or for that matter, any functional imaging experiment, relies on our ability to not only co-measure, but really code in real time and in real life the actual behaviors that are occurring. Outcomes. So we've done experiments using FNIRS, hyperscanning 2 or more people, and we looked at the outcomes of those. Was it an effective cooperation? How did the 2 partners rate themselves in terms of hierarchy and ability to influence the interaction and so forth? The products, moderators, socioeconomic status, sex, ethnicity, culture, etc. All of these are observable or measurable in real time, right? We can measure these things. I mean, it takes a lot to put these all together, and no one does all of these together, including us. But they're all measurable. They're all tractable. What about the stuff we can't measure? How about perception without conscious awareness? So we had one talk on visual perception, our last talk, and we have a visual pathway that's specifically designed for alerting. It's not for conscious perception, it's for alerting. So if I'm looking over here and someone claps their hands or just even makes a motion over here, it alerts me to look over that, and I'm not really consciously aware of that. It's a completely different visual system. Primary prediction paradigms. This is particularly well researched for visual stimuli, particularly emotion, so you can show sub-perceptual threshold facial expressions to people. And it's at such a fast rate that you ask them, what did you see? And they say, I don't know what I saw. I have no idea. Yet their brains will respond as if they actually saw the face. Do we have pheromones? I don't know. No one's proven it yet, but every other mammalian species pretty much does. We're going to miss pheromones, by the way, when we do Zoom. And baseline states and fluctuations may require sophisticated detection and processing pipelines. What do I mean by this? It has both to do with state and trait. Maybe we need to measure resting state networks with high resolution FNIRS prior to doing our FNIRS experiments to understand how the brain is ready or not ready to undertake a task or an interaction. There was this great paper that came out a few years ago in PNAS that showed that these endogenous fluctuations, this is an fMRI study, in the dopaminergic midbrain influenced your decision to gamble or take risks. But you weren't aware of this. This was just pacing along. And it had an effect on your decision making. So there's a lot going on under the hood. And what does that remind me of? We're getting to consciousness now. So if we look at the Earth from space, we go, oh, it's a pretty beautiful planet. We have land masses, we have water, oceans, seas, clouds, hurricanes. And it's a complex world looking at it from space. But in truth, there's so much more going on in the Earth. There's the magma, there's the inner crust, there's the mantle, et cetera, et cetera. And those are kind of potentially observable or measurable. And then there are things that require sophisticated sensors to measure, such as the Earth's magnetic field, which, by the way, has swapped itself several times in the history of our planet. There's some people who believe it's getting ready to do it again. So what do I think about neuroscience in the everyday world? I'm super interested in what we call interaction neuroscience. How do two brains interact both on an individual level and on a concurrent correlated level? Here's some of the areas that we're interested in. We have not done work in all these areas. Some of them we had: cooperation, collaboration, innovation, yes; competition, yes; persuasion, divergence, imitation, no. Although I think Yuan is going to do an imitation paradigm, hopefully, as part of her new work. Humor, we've done a lot of work, including some neuro studies. Creativity, emotional expression, empathy, etc. So this is what turns me on. This is what makes me excited about neuroscience in the everyday world. But it's not just me. Obviously, we're all here. We're very interested in it. And the general public has become more interested in this area as well. That was several years ago. This was just last week. Brainwaves synchronize when people interact. Of course, it's more complex than just synchronize. But people are very, very interested in this kind of dark matter, dark energy that is difficult to measure unless you design your study specifically to do it. I don't know how many of you have read this book, but it's also in popular fiction. So have you read the book Dear Edward or saw the Apple series? Anybody? Oh, it's really worth a read. There's this, so it's about it, it's based on a true story, but it's not the story of this particular kid. There was a plane crash and there was one survivor. It was a 9-year-old boy. And this series in the book is written by Anne Napolitano, based on a fictional survivor, solo survivor of a plane crash and what he has to go through and so forth. But it's really a very heartfelt, compelling book. And at one point, Edward wonders if the scientists who tend to the Large Hadron Collider are hoping to discover not only what happens in the air between two people, this is from the book, but how that pressurized air changes those people inside their skin. He hears a science teacher say, the air between us is not empty space. So, addressable areas of innovation research. I think we've got to get a whole lot better at detection and removal of artifactual compounds. We need to improve and miniaturize our sensors to remove interference that's occurring in the everyday world. We're a big believer in concurrent fNIRS fMRI for some of the reasons that Joy articulated, that it does go over well with grant reviewers, paper reviewers. If you say, hey, look, here we have the gold standard fMRI, and we're pretty much measuring the same thing with fNIRS. We're interested in it for lots of other reasons, including our deep brain inference method that we're trying with fNIRS. But I think it's a good thing to do. Multisensory approaches, multisensor, measuring moderators, outcomes, and lots of interaction neuroscience paradigms. So I say, let's go for the moonshot, and I think if this meeting is the beginning of the moonshot, that would be wonderful. Let's develop NIRS and other sensor technology and methods that are robust in the everyday world. And as I mentioned, it will require collaboration, not just among academics, but with industry as well. Now, I know David likes to show the future of fNIRS from, what's the movie you like to show it from? It's with Tom Cruise, isn't it? Yeah, anyhow. But I think this technology is possible, as shown to us, as shown to us from very realistic, future predicting entertainment. So one, Star Trek, does that ring a bell? Advanced NIRS, self-contained, and then the Riddick movie series, of course, where you have virtual reality type viewing along with NIRS. And I'm going to stop there. Thanks very much.
Thank you, Allan Reiss. So we have time for questions, roughly 10 minutes. Are there any?
Hi.
Yeah.
I share your opinion much with the DSM. And so I'm just wondering, what is your opinion on psychological scales? Is there any legitimacy to them? Have you seen that? I don't know, with like brain scans with FNIRS? If so, could you recommend some good ones?
Recommend some good psychological scales,
Yeah.
general ones. Well, you have to separate out robustness of the scale versus biologic construct validity, right? Whenever we actually see a significant association between a neuroimaging variable and a scale, I think it's a miracle or an artifact, because think about all of those steps that you need to get to, from genes to proteins to synapses to neuronal clusters to networks, and finally to behaviors and emotions. Sometimes we really do. We have to be careful about, correction for multiple comparisons and so forth. But it depends, obviously, on what you're studying. What are you studying?
I'm looking specifically at depression.
Yeah, I don't do those studies with imaging, but my inclination is to go for something simple, short, robust, and for which you can validate with some sort of observer scale, or something in the home, something with an interview. So it's not just reliant on one informant. Yeah.
Okay, thank you.
Yeah.
Hey, thanks for your talk.
You're welcome.
I had a quick question that I don't really know where.
The syphilis guy, right?
Yes.
Okay. That's such a great answer. I've never had that answer, but it's a great answer.
Yeah, I did actually read the DSM. I just didn't remember. But my question for you is actually about what do you think about the whole cortical blindness and schizophrenia, and lack thereof?
Cortical blindness?
Yeah, so it's a specific observation that's been like commented on a few times, and there's some debate because cortical blindness is very rare. It's 1 in 10,000.
You're talking about real cortical blindness,
real
like damage
blindness.
to the visual
not
cortex.
damage, congenital birth.
Oh, congenital birth. Okay.
Right?
Yeah.
So it's about 1 in 10,000, and schizophrenia is about 1 in 1,000.
Okay.
Or 1 in 100.
Yeah, it's 1 in 100 actually. Yeah.
And so we're owed at least one person who is cortically blind from birth and has schizophrenia, and they've never been identified. So there's been like population-wide studies where they've done this, and if you become blind later, maybe it's protective, but if it's from birth, cortical blindness, there's never been a person who's developed schizophrenia that we found.
I don't know what to say about that. I will tell you some of the more interesting, things do happen rarely, when you have rare conditions and you just do the probability multiplication, you got to find that person. Maybe they live in Peoria or something in a farm and they've never come to anyone's attention.
Yeah.
But I'll tell you some of my best hypotheses about something like this. So I studied one condition called Williams syndrome. Raise your hand if you've heard of Williams syndrome. Williams syndrome. So it is a condition where if you have it, it's a deletion, another deletion syndrome. You are unusually socially appetitive. So it's the opposite of Fragile X, which is kind of avoidant, and at least the stereotype of autism spectrum disorder, which is really not right, because there's lots of different styles and temperaments, and people get that diagnosis. But Williams, they just, the stories, we went to this national meeting of Williams syndrome, and we were early, so we had to sit down at the cafe, and within 5 minutes, we had 6 people with Williams syndrome surrounding us, talking, where are you from? Are you from Stanford? We'd like to talk to you about this and that. And so I thought Williams syndrome was all about faces, that they wanted to interact with faces. So 2 or 3 years ago, I had my first patient with Williams syndrome with congenital blindness from premature birth. I thought, okay, he's not going to be socially repetitive, and he was. It sounds like an interesting theory, but I wouldn't get too excited about it.
No. So if they've lost any kind of like if they have peripheral blindness, it's not protective.
No, it's
allegedly. But it's only the congenital ones that have been found, and it's like a population-wide studies from like Western Australia and for like over 70 years, and then there's some other European ones, but and some of the stuff goes and talks about like how visual cortex and role in dreaming and connectivity. So there's a lot of like
or
these features
attention.
that people
Like visual
to,
cortex is hugely important for attentional.
And it would change the whole way that the area would develop. But if you had like if you lost your sight, then it's already kind of developed a little bit in that direction. And if it's light sensitivity, then it's already sort of developed a little bit in that direction. It's all speculation, but
that's interesting.
Great talk, Allan. I really appreciate the overview of future directions. It's very important that we think about those issues collectively. I wanted to get back to the issue of funding because you didn't quite follow up. You noted that NIH is difficult, and you said you had some other alternatives, and I'd like to hear more about that.
I think we need to have a very interested foundation, large private donor that's interested in funding a consortium effort across multiple laboratories that has a review type process that's hopefully as unbiased as possible, but that does not have these increasingly difficult federal government regulatory compliance things associated with them. I mean, I found in how long have I been independently funded by NIH? It's a long time, since 1986. And I think it's at least an order of magnitude more difficult. And so I think for people who think creatively about how to advance science, and again, having chaired study sections, been on study sections, I don't think those people are rewarded. And I have to tell you, I've had the same reviewer that you've had for those first few comments.
I have them about every single time.
I swear to you, it's almost word for word. I told David that
word for word,
the same thing. So as even when you, by the way, even when you chair a study section, when you chair, there's only so much you can do to kind of unbridle the committee from thinking in a narrow way. This is terrible for me to say this to young investigators, but yet you kind of have to really play the game to get funded. And I think young investigators have a little bit more leeway than more these days, which is good, the more senior investigators. But I think we need a huge infusion, some sort of endowment, something to kick this area off.
All right, I co-sign that.
Okay, good.
So now I'm wondering, you were talking about the variability of the disease and the diagnostic and the scales and not the trust.
The DSM stuff.
Fine,
yeah.
Fair enough.
Yeah.
And probably I'm just interpreting this incorrectly, but how is FNIRS helping you then with your studies?
Yeah. So you can see we don't study a lot of those DSM disorders. And I'm very careful about advising people if they do. Now, one of the things I was talking about at lunch was about PTSD. I kind of reserve a little corner of less speculation about PTSD, not because I think it's a homogeneous entity from the standpoint of pathophysiology, but I think it's a common denominator endpoint if you have sufficient stress over long enough duration and you are susceptible genetically, neurobiologically. Pretty much everyone comes to that same point. Okay, not everyone, but most people do. So I think that's one exception. I would have told you 20 years ago, Tourette syndrome would be another exception. Bipolar disorder would be another exception because they're so familial. We should have figured them out by now, but we haven't. So that's why I study Fragile X. I study diabetes. Diabetes is complicated, but at least we know the parameters, the glycemic parameters that affect outcome. So it's more tractable. And my feeling is, for me, if we can't figure that out, there's no way we're going to figure out ASD, ADHD, schizophrenia.
So then.
Okay, so let me give you one more answer.
Yeah.
You can look at traits, cognitive parameters that tend to cross species, like inhibition. That's a good one to study because it's very definable, it's very measurable, and it turns out it's much more genetically heritable than, for example, ADHD. That's what I'd be going after.
Got it. Makes sense. Now, depression has been a big target application for FNIRS.
Yeah, I know.
Maybe like if you like the single most published application for FNIRS.
Yeah.
And I think Japan, for a while at least, maybe they revoked it, had approved FNIRS as like.
Yes, they did,
Right.
as a way to differentiate depression from schizophrenia.
Right.
Which is kind of, sorry to say, ludicrous to begin with, because those are both constructed constructs for the field, and you can just differentiate them just by the diagnostic phenomenology. But as it turns out, they're not necessarily so distinct from a genetic predisposing point of view. Yeah.
Sorry, I have this really simple sort of worldview of maybe depression that I understand from the literature, very naive readings. It's just you have to normalize hypoactivity in the
Frontal cortex.
Yeah,
that's what they say about schizophrenia too.
Right, and
so yeah,
and so like from that perspective, it kind of you can measure the effectiveness of treatment maybe to see if you're getting that renormalization of brain activity. And that's a nice simple story.
If someone could show that to me, right, I would be a believer. I would join that religion. But I don't think it's happened because there's so many, we already know there are dozens and dozens of different risk factors for developing a depressive disorder. At least dozens and dozens.
I have a related question, sort of inspired by your comment that it's almost a miracle or an artifact if NIRS is used and there's a correlation identified to something from the DSM.
Or just even a scale, forget the DSM.
Or to a scale.
Yeah.
Okay. So in conversation earlier, I was discussing with you some global health application.
Yeah.
So across cultures, and I know you said you've done some work in Costa Rica. So I'm wondering, I imagine that increases the challenge of studying psychopathology and possible universality of the metrics across cultures with NIRS or with EEG. And I wonder, do you think that's near impossible, or what solutions do you
No, not
see?
at all. So for example, in our study in the Central Valley of California, looking at the long-term and even in utero exposure to insecticides, I think we have 500, is that right? 500 participants, all of whom got NIRS, most of whom have measurable levels of insecticide exposure. So you got to get big study samples for epidemiological studies. Costa Rica was a smaller study. They looked at organic farming communities versus standard use of insecticides, and got enough power that way to see differences.
Okay. So it's not impossible.
No.
And I do see studies that are implemented in different countries, especially for depression, do show similar results, but aren't necessarily comparable to one same scale.
Yeah.
Is the conclusion. Okay.
Yeah. It's not that I don't believe in depression. I do. I mean, I treat depression all the time. It's just that as a phenomenon, it does not have robust genetic or biologic construct validity. It's measurable, it's replicable, people can arrive at the same diagnosis. We can make some of those people better, maybe about 50 to 60% with psychotherapy and or medication. But, it's not like having pneumococcal pneumonia.
Yeah. Thank you.
You're welcome.
We have one question from the audience and then we move on. This will be a good one to end on. So this is from Ray Hane, and they ask, so you mentioned that you will explain what consciousness is not. Do you mind?
Yeah,
explaining that a
consciousness,
bit more?
consciousness, I think, is looking at the Earth's atmosphere from space. And there's so much else going on under the hood. That's what I think consciousness is. So all that other stuff, okay, magnetic fields, crust, everything else, that's not consciousness. And I think consciousness is probably overrated as something that's really, really important to human behavior. Personal opinion.
Thank you very much.
Thanks.